So I think we'll get started. I want to welcome everybody, thank everybody for coming. At least today wasn't snowed out, although Ed said the traffic coming in was not good.
I was glad I left as early as I did.
So I'm absolutely delighted to introduce Ed Scolnick today for our conversation with the scientist. And I would say I'm delighted for two different reasons. First, these conversations. They have been great. We have some of our conversants in the audience today. The precedent sets a high bar, Ed, and they've been enjoyable and they have been very educational in terms of both a history of biology and really a history of biologists. They've been very personal. And I basically will go to every single one I can possibly go to because I've just really enjoyed them. So that's number one about what makes this special. Number two about what makes this special is Ed, because Ed is special. And I actually had the privilege of introducing Ed once before. It was 4 years ago. It was for a dean's lecture that Mark Kastner had set up. And Ed gave a lecture for this Dean's Colloquium, and I have the title here, The Power of Basic Science Applied to Medical Progress, Past Examples and the Hope for Schizophrenia and Bipolar Illness. Okay. It was a great lecture. It was science and personal based. If you want to watch it, you can watch it. It's on the MIT video site, which you get to by just putting in video.mit.edu, and you can find the lecture. For that lecture, I was asked to give an extensive 15-minute introduction. So if you haven't had enough of me, you can get the introduction there. And if you want to fast forward through that to get to the real stuff, you can do that too. The simplest thing I can really say about Ed is that he is literally a legend in his own time. And he's known through academia, he's known at NIH, he's known in the pharmaceutical industry, and he's known for a variety of different things. And when I gave the previous introduction, I asked a number of people who I knew had known Ed for a long time, what they thought of him. And there was one comment that was really telling. I asked somebody who used to work for Ed at Merck what he thought of Ed, and Ed doesn't look too nervous, and that's okay. The response, and this is a verbatim response, I am an unabashed hero worshiper when it comes to Ed Scolnick. Okay, that's a real quote. And I think it doesn't get better than that. I've known Ed now for, I guess it's about 25 years. I knew Ed before he went to Merck, when he was a researcher. Ed trained as a physician. He then became a research biochemist. He worked on the mechanism of protein synthesis, and then turned to tumor virology and made an incredible and very well-known discovery, namely RAS, as a viral oncogene. Now, of course, we know RAS locally from the work of Bob Weinberg that showed that what Ed found in a viral genome also has a counterpart in the human genome. Very important in cancer, and it was actually at that stage we started talking because we were working on a RAS gene in C. elegans, and Ed kept calling me every month saying, what do you know now? And I kept saying, not all that much more than we knew a month ago. But
Yeah, I had forgotten all about that.
yeah, no, I remember the calls. Ed then moved to Merck. He had an exceptional career there. He was president of Merck Research Labs, 22 years at Merck. And amongst many other things, he helped develop the breakthrough therapy that has been used for AIDS. He then made another transition and came here to the Broad Institute. And at the Broad, he's become a leader in applying genomics basically to the field of neuropsychiatry. And he was the founding director, that means also fundraiser, for the Stanley Center, and has had really a pivotal impact on our local community here. And I have to say personally, I've been very delighted to have Ed close by since he's come here. I've never failed to be impressed by Ed, delighted by my interactions with him, and I have to say that I am very much looking forward to his remarks, and I will not take another minute of his time. Ed, welcome.
Thanks.
Thank you.
Well, this is a very different kind of interaction or talk than I've given in many other venues. And I was told it was supposed to be a personal history with enough information that students could glean something from it besides just knowing more about me. So I'll try to do that. I grew up actually in Boston, in Dorchester. I was born in 1940. When I grew up in Dorchester, it was a very nice community, quite safe to roam around in, and very supportive, but extremely insular. I basically grew up in something that might be called a ghetto environment. I had almost no exposure to anything outside my immediate family and the local schools I went to until it was time to go to college. I was fortunate that at the time I went to grammar school and high school, and I ended up going to Boston Latin School at a time when entrance exams were not necessary. I went to Boston Latin School, and at the time, if you did reasonably well at Boston Latin School, you were almost guaranteed that you'd get into Harvard if you applied to go. And I applied, and I was admitted. I wasn't really sure what I wanted to do, and I liked science. Thought I would go and be a physician because the environment I grew up in a Jewish household in the 50s, really, that was kind of the goal for nice young men growing up in that environment. And that was all I had in mind for my professional life at that point. I would make a couple of remarks about my first years at Harvard and the difference at MIT. I had an enormously difficult social adjustment to Harvard in my first few months. It was such a different social world and so diverse compared to what I grew up in and socioeconomically from what I grew up in, that I found it extremely, extremely stressful. And I was always highly motivated to do well in academic studies. And I decided finally in around November of my freshman year that I had to do something different or I might really decompensate. And so I looked around the community for what kind of activities to get involved with to try to find a part of this massive university, a smaller section of it, a smaller cut of it, where I actually could feel I belonged to something. Because I never felt like I belonged to Harvard University, Harvard College. I felt very out of place. And I ended up pledging for and joining the Harvard radio station, WHRB, which still is on the FM dial. And that was actually transformational for my life in college. It enabled me, it forced me to be organized because of the amount of time spent at the radio station versus having to study. And it did give me a social focal point for a smaller organization where I could feel I belonged to something and was comfortable with the people that I was with. And I ended up doing radio broadcasting for news, sports, and special events at Harvard, which again was a transformational event because in learning how to broadcast on the radio and being taught what you had to do to communicate with people who were listening in, and who can only hear what you had to say once, as opposed to reading the newspaper where you can reread stories, it really helped my social growth, it helped my communication skill growth, and I made an enormous number of terrific friends, some of whom I've stayed in touch with until a couple of them passed away. And it enabled me to relax and feel very comfortable in the university setting. And I decided to major in biochemical sciences. And the second event at Harvard, which is very important and has, some of you know, a real MIT connection, was Harvard College had a so-called tutorial system for students. I don't know if it still has that. And what that meant is you met on a monthly basis with a professor somewhere in the Harvard system who you met with and discussed and read about topics in science outside what you were taking in your courses. And I was fortunate to have Boris Magasanik, who at the time was a professor in the microbiology department at Harvard Medical School. So he was part of the Harvard tutorial system, and I was fortunate that Boris ended up being my tutor. It took me a while to appreciate Boris. He was so much more sophisticated than I was. And to appreciate what he was trying to teach me, which was how to think in science as opposed to just memorize. And I met with him regularly, and in my junior year, or just before my junior year in college, and things had begun to go pretty well for me, Boris moved to MIT in the biology department. Salvador Luria, who I think in modern times really built the biology department here, recruited Boris from Harvard to MIT to help build MIT biology. But Boris stayed as a tutor, and in my junior year in the fall of college, so this would be 1959, 60, he said, I'm moving to MIT, and you like science. I'm going to be teaching a course in the spring with Salvador, and you should take the course. So I said, how do I do that? I'm a Harvard student. And he said, well, it's easy. You just go to the registrar's office at Harvard and you tell them in a few forms. It's very easy. And take the bus down Mass Ave and come to our course in the spring. And I said, well, that would be great. I really enjoyed the little bit I knew about bacterial genetics and what Boris used to do with me in tutorial. And this is a time in science, as some of you may know, where it was really the fomenting time in bacterial genetics and bacterial physiology, where these basically a lot of mathematically gifted people after the war went into bacterial genetics and phage genetics because pre-cloning days, they were genetically manipulable systems where people could start to understand cell biology and how cells worked, even though it was mostly E. coli cell biology. And I thought that was really, really interesting. I had taken a course at Harvard, either my first or second year, I think it was my first year, taught by George Wald. It was a mandatory general education course. And like all freshmen at college, particularly people who came from a pretty insular background, I was trying to figure out what life was all about. And you'd spend your late evenings with your roommates discussing the philosophy of life. And none of that led to anything particularly profound. And one day I came into George Wald's lecture and he gave a talk about DNA and the double helix and what was known at the time, which is 1958 or roughly. And I heard that lecture, and Wald, as some of you know, was a fabulous lecturer, just lucid and excited about science. And I said, oh my God, that's how I can learn about life. And literally, it was one of the transformational events in my education. And then Boris had me take this course with he and Salvador, and that was a second transformational event because it was completely different from any course I'd ever had in college at that point. because instead of making you memorize things and certainly integrate them to a certain degree, it was all experimental biology. They either gave you experimental results and asked you to interpret them, or they gave you a biological problem and said, in bacterial physiology, said, how would you design an experiment to try to do this? That was stressful. I had never had to do that. And I really struggled in the course for a few weeks. And I was fortunate to sit next to, in the course, another person many of you know, Sam Latt, who was an MIT student, an incredibly gifted person. I think ended up at Children's Hospital as a professor, died prematurely, unfortunately. And Sam befriended me and spent a little time after class explaining to me what was really going on in the course, and I managed to pass the course. But I loved the material. I mean, having heard Wald and then Boris and Salvador teach this course on bacterial genetics, all the classical experiments, and I decided for sure that I wanted to somehow do work in that area of biology because you really could potentially understand how cells worked, how the body worked. And I struggled as to whether to go to graduate school and get a PhD, which Boris encouraged incessantly, relentlessly, or to go to medical school. And I just didn't have the courage to go to graduate school. I thought going to medical school was safer, and my parents pushed me a little bit to do that in addition, so I ended up going to medical school, and I ended up going to Harvard Medical School and having survived an interview by Daniel Funkenstein. I can't remember his first name. There was a psychologist who used to interview all the medical students for Harvard. His name was Funkenstein, and he was notoriously difficult. And he would put you in very awkward situations. So I grew up in Dorchester. I went to college at Harvard. I went to wanted to go to medical school. I'd never been out of the city to travel. So I remember he had the reputation of nailing down windows and asking students to open them and seeing how they work under stress. He didn't do that to me. He said, you grew up here, you went to school here, you want to go here. Don't you think you're pretty narrow? And I said, well, you missed something. I was born at the Boston Lying In Hospital, and I just feel comfortable in this area. So they ended up admitting me, and I found Harvard Medical School a magnificent intellectual environment. I loved it. I was happy I had made that choice. I really was interested in the science and biology of medicine. I had done some research in my senior year at college with someone Boris had put me in touch with, Ed Lin, who was a professor in the, I think actually in the physiology department in Harvard Medical School on a bacterial genetics physiology project. And it had gone all right, but not swimmingly, I would say. But again, the atmosphere I found just wonderful. Ed Lin used to have once a week evening sessions with the medical students who were working in his lab, where they would bring in Chinese food or some pizza or whatever, and one of the students would have to get up in front of the group in a very informal atmosphere and talk, and there'd be a couple of hours of questions and dialogue. And again, I just found the whole atmosphere of doing that enormously stimulating and tremendous fun. So I really wanted to do that. I wanted to do research in my life. What I had done in medical school was only fair and really not a great experience. And so I was going along during my medical school life, not quite sure what direction to go in. And late in my junior year, early senior year, this is in the mid-'60s, the Vietnam War was enormously heating up and a miserable situation, as many of you know. And I realized that the odds were I was going to get drafted when I graduated or finished my internship and potentially get shipped over in something called the Berry Plan to take care of soldiers in a war situation. Clearly, I didn't particularly want to do that. And at the time, when there was a draft, there was still a mandatory draft. It was known that if you got into the so-called public health service and went to NIH, that would serve as your 2 years mandatory draft requirement. So I went to the dean's office at Harvard Medical School and said, I'd like to spend the last 6 months of my 4 years here doing research because I wanted to be able to do enough to potentially get into NIH. And they said, sure, you can do that. You clearly have a good track record. And the only problem is you haven't had OBGYN yet. And you're going to have to pass part 1 of the national boards when you graduate medical school. So if you think you can pass without taking the course, we'll let you do research for 6 months. And so I bought a little book and I read it and I managed to pass the test. I knew I was never going to deliver a baby, so I was not worried about that at all. And I worked for a man named Dan Dakin, who I think is still alive and a professor at one of the VA hospitals, at least that's where he used to be. And at the time was at the Beth Israel Hospital. We worked on platelet biochemistry. I was interested in atherosclerosis at the time. And that went, again, kind of okay, nothing great. I liked it. I wasn't particularly good at it. And I had basically given up the thought that I could do research. And, but the people who had mentored me, Ed Lynn and Dan and others, thought I still had potential. And when I applied to the National Heart Institute, I was accepted. In the meantime, I had gotten accepted to be an intern at the Mass General Hospital, which was a shock. I never thought I'd get in there. It was so different from what I had grown up in. It was a bastion of white Anglo-Saxon Protestant medicine and culture, not anything like my background. But I remember the interview I had when I was interviewing for the internship. And as I said, I really loved genetics and biology. And we were a group of students sitting around a semicircle being interviewed by a series of professors in medicine. And the question they asked the group at one time, so you're in a group environment and you're obviously trying to show off your compared to your colleagues who are competing to be accepted as interns. And yet, you know, you didn't want to be a jerk and really try to be too pushy. So they asked a question about childhood chronic myelogenous leukemia. They asked a question about CML or I think, or some leukemia like that. And they asked the group, well, if you wanted to start to explore the causes of this and who would you go to and how would you go about it? And I used to follow the literature a little bit in genetics, and the other students in the group chimed in and they talked about a professor at Children's named Park Gerald who was interested in leukemia and genetics. And I just didn't say anything. And then when they kind of stopped, I said, yeah, well, that's very good, but there are these two guys at the University of Pennsylvania, Noel and Hungerford, who have described something called the Philadelphia Chromosome and a two-hit theory of cancer. And I went on and on about that. And I was absolutely convinced that that's what got me into the MGH, because they could see that I had some breadth to what I knew about. So I was accepted to the MGH and then accepted in principle to go to the Heart Institute. You learn this in the beginning of about the time when you're beginning your internship at the MGH. And I loved being an intern, even though it's exhausting, and a first-year resident. I really learned how to be a careful, scientifically oriented person and scientist in the first few months I was an intern, for two reasons. One, you're so responsible for people's lives in a stark way that you appreciate the weight of that responsibility and how much you really have to be careful and respond. And I was really lucky. I had an assistant resident at the time named Bob Snodgrass, who I'm told is still alive somewhere in the Midwest practicing medicine, or Mid-South. Snodgrass was a great guy, totally unpretentious, and took care of his patients like nobody I had ever seen or met at that point in my life at the MGH. And I realized what he did was what was necessary. So I became a super compulsive, dedicated intern. And it was great. I loved what I was doing. And then toward about halfway through the internship, maybe in the spring of that year, which would have been 1966, 65 or 66. No, I'm sorry, I'm off here. Yeah, 66. You went to the Heart Institute to interview with lab chiefs to do another matching for which lab you'd end up with. And so I went down, and it was a two-day session, I think. I went around and met a lot of people, and I wasn't really interested in most of the people I met. I didn't really want to work with them. There were two people I was really interested in. At the time, there was a biochemistry laboratory in the Heart Institute. It was overseen by Earl Statman, who was just a giant in classical biochemistry. Many critically important contributions to both cell biology, biochemistry, and bacterial biochemistry. The regulation of an enzyme called glutamine synthetase was probably one of his finest things, and it was just such a complicated system, and he worked it all out gorgeously with a combination of biochemistry and genetics and bacteria. And one of the people in his laboratory who had a section was Roy Vagelos, who was studying lipid biochemistry. I met with Dr. Vagelos and told me what he was doing, and I had been interested in atherosclerosis, and I said, boy, this is really terrific. I'd like to come work for you. Would you take me into your lab? And he said, I think so. I want to do a little more checking on you, but yeah, potentially you can count on this. And then I went to, but he said, there's one thing, and I'll keep you informed. I've been negotiating to be chairman of biochemistry at Washington University of St. Louis. Corey's old position, again, a giant in biochemistry. And if I take it, don't come here because you won't have a lab to come to. So I said, okay, let me know, and he did. He subsequently called me at home like a night or two later, said, I've decided to take the job, don't come here. And then my last interview in the Heart Institute was with Marshall Nirenberg, as Monty knows, one of my tremendous heroes, and I think Monty's heroes. And so I knew what Marshall had done. I had seen his paper on polyphenylalanine and understood the excitement in the genetic code, because all of the stuff that was talked about in the Luria Magasanic course was about bacterial genetics, and was the code Triplet, was it 4 bases, 2 bases, 6 bases? How could you figure it out? So I sat down, started talking to Marshall, and I was mesmerized. I was absolutely mesmerized. It was obvious this was a very exciting person. The work was spectacular. It was interesting. And we talked for a long time, more than an hour, just about what he was doing. And I said to Marshall at the time, I'd really like to come here if you would have me. And he said, no, I've had a lot of MDs come, and you've got a good pedigree, and I'd be happy to have you come. I said, but Marshall, I have one question for you that really bothers me. I'm sorry to ask you this, because you may think it's a ridiculous question. He said, no, go ahead. I said, what you're doing is great. this field, I know from the outside, is fiercely competitive. How do you deal with that? How do you deal with that? I'm not sure I'm psychologically capable of handling that kind of competitive pressure. And he smiled. He just sat there. It was beautiful. I can never forget the smile on his face. And he said, look, Ed, most of science fails. It's 99% perspiration and 1% inspiration. If you're going to go into science, you have to pick a problem that if you do succeed, people will care about. And I said, wow, that's pretty amazing. I said, that's really true. I said, okay, I'll take a chance. What have I got to lose? I'm coming here for 2 years. I can go back to medicine. If it doesn't work out, I'd like to come. And he said, okay, we'll accept you. Send in your matching list. So I did. I matched with Marshall's lab and came in the summer of 1967. I had no idea what was going on in the lab or how to do research when I got there, and I realized that instantly, absolutely instantly. None of the technologies that were being used, I didn't know how to do any of it. I mean, nothing. And stumbled around, and Marshall said that he was going to work in neurobiology, and I ended up working more directly with Tom Caskey, who was a sort of senior position in his lab. And at the time, the one important remaining problem in protein synthesis was the termination signals and the biochemistry of termination. And so we worked with Tom, and Marshall taught me what I had never been taught in college or medical school. That is, how to do research, how to think about it, how to think about the execution of it, how to think about the design of experiments. Marshall was a true genius in more than just the intellectual part of how thinking of projects to work on and how you approach them. He was a genius at teaching you a system of doing science experimentally that, and I've said this to people in the Stanley Center many times, that guaranteed that you could never have irreproducible results. And I never had been taught that by my professors at college or medical school. And if you want to lecture on that, invite me back and I'll tell you how to do it. But you can ask Monty too, because he still uses the same system. And that really transformed my life in biological science, because I realized all the stupid things I used to do when I did research in college and medical school, and how I was my own worst enemy. And we struggled for about a year to try to figure out an approach to the termination problem. It was known at the time that of the 64 triplets in the code, there were 3 that were unclear of their function, except that they probably were responsible for protein termination as the chain finishing and falling off the ribosome. And there were UAA, UAG, and probably UGA. No tRNAs had been found, natural tRNAs for UAA and UAG, and the UGA one was not considered that important at the time, and it was ambiguous. So we worked, and Tom led the group for several months trying to figure out how to approach this. And in about the middle of the year, a young assistant professor, someday to be a Nobel laureate at Harvard Medical School, named Mario Capecchi, who was at Harvard Medical School at the time, published a paper in PNAS using a phage system in which he had ribosomes programmed with a RNA phage messenger RNA, I think it was R17. And when he programmed that with partially purified factors and just ribosomes, the polypeptide chain translated on the phage message was made and stayed on the ribosome. When he added some supernatant factors from the bacterial extract, he had a sucrose gradient method where you would spin down the ribosome and then assay the stuff that stayed up. You could knock the chain off the message, and that it was related to the UAA termination codon. So it was something in the S100 that it was a very cumbersome assay, but it was a beautiful experiment. It was just gorgeous. It opened up the field, but it was a very cumbersome system for trying to go further and figure out really what was in the S100. Tom Caskey had a brilliant idea after that came out, in which he picked up on the triplet binding assay that Phil Eder had developed in Marshall's lab for codon recognition, in which he created a dinucleotide codon, AUG terminator codon, bound to ribosomes, and with, on the complex, formylmethionine tRNA. So formylmethionine is blocked at its N-terminus, so it's like a little peptide. And you could make that complex in vitro, and importantly, you could freeze the complex. So you could make gobs and gobs of FMET tRNA bound to the ribosome with the initiator codon and terminator codon. And then he worked out an assay where if you hydrolyze, because Kopecky had shown this work, S100 could hydrolyze the peptide. It hydrolyzed the peptide, simple extraction procedure, because it was hydrophobic at that point, and you had a very rapid assay. It took about 10 minutes to do the whole experiment, and then process your samples, put them into the scintillation counter. The label was in methionine. So this was in the late spring of the first year I was at NIH, and excited but not feeling particularly great, because we hadn't discovered anything. And I was still thinking I'd go back to medicine. I was going to apply for fellowships in infectious disease and hematology, because they were kind of molecular at the time, and I thought that would be what I would do. Tom went away for vacation over Memorial Day weekend in 1968. And he left myself, another postdoc named Dick Tompkins, and his trusty technician, Teresa Carrick, who I met at an honorary event for Marshall last summer. She's still alive and looks great. And we were supposed to fractionate the S100 using the assay and figure out what the factors were in the S100. So Dick Tompkins ran some of the assays, I ran some of the assays, and Teresa ran the termination assay, because she was Tom's personal technician. And so we ground up a kilogram of E. coli, and we made the S100, and we worked like the dickens all day, and we fractionated everything out on a column. I think it was a ion exchange column, and we looked at the data late in the afternoon, and it was very clear we had all the regular protein synthesis factors. They had come off the column, they had their peaks, and Teresa brings us the termination data, and there's a big peak of termination factor coming off the column, much later than any of the other known factors, so we knew we had something unique. And the peak goes up, and it's about right here coming down. So Dick had to leave. He had a family at that time, and a child in addition to being married. And I said, Teresa, we can't leave it here. Let's just go out another 40 tubes every third or fourth tube, and we'll pool the peak, we'll put it away, we'll come back and study it. I says, okay, they're going to be here after 5. And she said, oh, sure, no problem. So she runs the assay, which doesn't take very long, and comes back and shows me the data. And this was the changing event scientifically in my life. because it produced a dopamine rush that was like nothing I had ever experienced. And I was addicted forever to science, totally addicted. Goes up, comes down, flat, second peak, up and down. 2 peaks? Not in Kopecky's paper. He had no peaks. So I said, Teresa, it's late, but do you mind staying? You know, because she had run all the assays. I said, no. I said, take the peak tube from peak 1 and the peak tube from peak 2 and run it against each of the 3 triplets. So she said, okay, good. She does this, and I'm sitting and waiting, and she comes back, and it is the clearest result that one could imagine. Peak 1 terminates with UAA and UAG and not UGA, and peak 2 terminates with UAA and UGA and not UAG. And I say, oh my God, we've got the termination factors. There's codon recognition here. So we scurry around together, and everything's been run in duplicate, and there's just no ambiguity. There's no p-values. There's nothing. It's just black and white. I didn't need Mark Daly to calculate the p-values. So we put all the stuff away so we can study it. It's very late on Friday, maybe 9:30, 10 o'clock. We're both excited. And Marshall is such a great guy. I call him up at home, and I say, Marshall, Dad, you got a minute? Yep. Let me tell you what we just found. And I tell him the result. The first thing, this is classic Marshall Monty, he says, what are the controls? And I say, Marshall, you don't understand. The peak tube is the control. It's inherent in the experiment, reciprocal experiment. And he says, oh, wow. And then he won't let me off the phone. He's got me there for another half hour. And I actually, it was great. I went home, my wife was furious, I was so late. We played a trick on Tom. We told him nothing worked on Monday morning. We hid all the notebooks from him, and then, you know, it was all great. And at that point, I decided I wanted to do this. And it was such a gratifying experience. And I do believe what I said. There is a dopamine rush. You get such a high out of that it is almost like an addiction, or if it's not an addiction. And I just wanted to keep doing that in my life. I had enough confidence at that point to do it. So Marshall said, well, if you want to stay in the lab and be a staff person in the Heart Institute in Tom's section, you can do that. And I said, oh, that'd be great. It's terrific. My wife's pregnant at this point, and we're looking for some stability in our life. And we decided to stay at NIH and in Marshall's lab. And then, after about a year, most of the project was over. There was still work to do in the mammalian system. And another kind of just interesting anecdote, when I had been an intern and resident at the MGH, when I was a first-year resident in medicine, Joe Goldstein and Mike Brown were interns. And I had very little interaction with Mike, a little bit, but Joe turned out to be my intern. The ARs always taught the interns. So Joe was my intern. It turned out like 2 or 3 times in a year by chance. So we got to know each other. We're friends. And he called me in the spring of my second year in Heart Institute, and he said that the lab he had applied to the person was leaving to go to some university. He didn't have a lab. Could we help get him into Marshall's lab? So I went down the hall and talked to Marshall, and Marshall said, good, it's another smart MD, but we have no space. I said, well, we'll squeeze him into our 200 square foot module. And so we worked together for a couple of years and again, remained extremely close over the years. So I stayed in Marshall's lab for about another year. The project was kind of coming to an end. We were doing the same thing in mammalian cells. And I'd come to realize what every young person in science, I think, struggles with is your own identity. I realized that if I stayed in Tom's section forever, as much as I liked him and liked Marshall and liked the lab, I'd never really have my own identity in science. And I didn't like that. I decided I wanted to have my own identity. So I didn't know quite what to do. And everybody in science who I talked to at that point said, well, phage genetics was the key field for understanding bacterial physiology. And mammalian cell biology now is starting to think about using animal viruses in the same way bacterial geneticists used bacterial viruses, because this is pre-cloning days. Remember, they were littler organisms, and they could be manipulated a little bit genetically. And so you had the chance, much more complicated, to use them to understand mammalian cell biology. So I said, a lot of smart people thought this way, sounded good. And Cold Spring Harbor had these summer courses, which it still has, and it was teaching a course that summer in animal virology. So I signed up for the course. My wife came during the summer. Our second child was born at Cold Spring Harbor that summer, was impregnated. And there were some great people in the course. Ann Skalka, Phil Leader, a couple of people from overseas. It was a wonderful environment, and the course was taught by Phil Marcus, used to be a professor at Connecticut, I think, of microbiology, and a couple of other people, and it was fun. I learned how to culture mammalian cells, I learned how to work with animal viruses, and I decided this is good. I'd like to do this. I wasn't sure whether I wanted to do it in a research environment or a medical school environment, doing some clinical medicine, some research, and I started looking around for opportunities to do this. People at the MGH, a couple of people at Stanford, and at the time, so now it's 1969, 1970, the country, Richard Nixon et al., decided to have a war on cancer. And I had learned about tumor viruses as part of this course. They had had Howard Temin, one of the fathers of RNA tumor virology, come to the Cold Spring Harbor course and give a talk on RNA tumor viruses. Mysterious, but clearly important. And there was a good lab at NIH at the time, because I was still part of Marshall's lab, studying tumor viruses. It was run by George Tadaro, who had done some very nice cell biology. He and Howard Green developed contact-inhibited cell lines from mouse embryos, and they were clearly regarded well and were close by. So I went to interview with George and said, I'm interested in animal virology, tumor virology. Could I come to your laboratory and give up my tenured job in the Heart Institute and be a senior fellow? And he said, yeah, you've got a great background. We don't have anybody here with molecular background. We'd love to have you here. So this is like May or April. And so I started trying to learn a little bit more about tumor viruses. And there was a lecture given at the National Academy in Washington by Saul Spiegelman, who was a famous and very productive and creative and showy bacterial biochemist, or I'm not quite sure what you would call him. So I went down to hear his lecture on the replication of RNA tumor viruses. And I listened to it, sat in the way up in the balcony in the auditorium, and I heard the lecture, and he talked about the RNA-dependent RNA polymerase of Rose sarcoma virus and how that explained everything. And I had been reading, and I said, doesn't make any sense. Doesn't explain the DNA requirement for replication. But that's about all I could say at that point. And at that point, July 1st came, I told Tom and Marshall what I was doing, and they said, great, your choice. And I joined George's lab. And I've been there about a week learning how to do cell culture. When George wanders into the fellow's office, where myself and Stu Aronson, Wade Parks, we all share this big office. And he says, I just got a call from David Baltimore. And David says he's just discovered an RNA-dependent DNA polymerase in Rose sarcoma virus. And he wants to come down to get some murine retrovirus to be sure that this is general, and he's going to come beginning of the week. So boy, this is really exciting. And so David Baltimore arrives in our contract lab in Northern Virginia. We're introduced and tells us about his discovery. We're going around touring George's lab. It's a big, big facility. And while we're going, I said, Dr. Baltimore, you know, I'm just new here and I don't know that much about the field, but you know, it looks like your discovery explains provides the basis for the replication. I said, from what I know, it doesn't really explain the cancer part, does it? He said, no, that's completely mysterious still. So when he left, I went to George Tadaro and I said, George, I want to study the cancer part because the replication part is now known. And he said, well, you can do whatever you want. We need you to help us set up certain assays so we can look for human retroviruses. As long as you do that on your own time, you can do whatever you want. And I said, great, good. So I did. I helped them set up their various assays. And then started trying to figure out how to approach the cancer part. Now, for those of you who don't know anything about the history of RNA tumor virology, the major beginnings of that field really came from the chicken world, the chicken virus world, Rous sarcoma virus. And they were extremely fortunate in that they had a single virus, a unit virus, that both replicated and transformed. You could clone the virus and inherent in a single virus with both properties. So they could manipulate the replication genetically, and they could manipulate the cancer part, the oncogene transforming part. And Peter Vogt, Peter Duesberg, and there was another guy at Berkeley who, I can't remember his name now, made the first temperature sensor, Martin, right. What was his first name, do you remember? Steve Martin, yeah. He made the first temperature sensitive mutant of Rous sarcoma virus showing that transformation was a biochemical function of some protein. And then Duesberg and Vogt did these magnificent experiments where they showed that starting with a virus particle, you could isolate so-called transformation defective mutants, which had lost the oncogenic properties. And Duesberg and Vogt together fingerprinted these with RNA fingerprinting, again, pre-cloning, showed there was some oligonucleotide spots missing in the transformation defective virus. So it was pretty clear that there was some kind of oncogenic function coded for by the virus. In the mammalian system, we weren't so fortunate. All the viruses that transformed in culture were replication defective. And Stu Aronson in George Tadaro's lab with George had shown that you could isolate the oncogenic virus, the transformation defective virus, in so-called non-producer cells. You could get endpoint diluted foci where the foci grew up and no virus was coming out, and then you could add replicating murine helper virus, a retrovirus, rescue it and transmit the foci. So all the viral stocks that transformed had two viruses in it, a replicating component, transforming component. George's lab was interested in the genetics and predisposition to human cancer, and so they were using, by pure chance, a virus called the Kirsten sarcoma virus to look for genetically susceptible human fibroblasts. And the reason they chose Kirsten virus was you could see the foci on human fibroblasts better than you could with a number of other oncogenic viruses that were available at that time in the murine system. So naturally, I started to work on Kirsten sarcoma virus. And there was a Kirsten leukemia virus, basically a replicating helper. So about the only thing you could do, because it was too complicated to do fingerprinting, you could do hybridization experiments back then with the technologies that were available. And it was clear from the chicken virus work that there was something called an oncogene. So we started making stocks of the pure helper and the mixed virus. There was about a 1-to-1 ratio in the mixed virus. And making cDNA from one, cDNA from the other, using the endogenous enzyme of the viral preps, and then cross-absorbing and looking for what was left. And we did these experiments, and it was very clear that the sarcomavirus stalk had sequences in it that were not, is this too long? Am I running over? No? Maybe too long?
Yeah, I'm trying to ask for guidance. What time?
Yeah, what time? Noon?
So, no, 5 minutes.
5 minutes. Okay, we'll end in 5 minutes, and then you can ask questions.
Then we'll need a part 2.
Then you'll need a part 2. So I'll finish the story, which is worth finishing, I think, you'll see. And there is a lesson in the story, which is why I'm telling it. It was very clear the sarcomavirus stalk had sequences in it that were not in the leukemia virus stalk. Did this experiment dozens of times. No idea what to do with it at that point. Absolutely no idea. And one morning, in my home on a Sunday morning, I was literally in the bathroom reading a textbook, sort of historical volume, written by Ludwig Gross, one of the fathers of tumor virology. And he describes all the tumor viruses that have ever been discovered, DNA and RNA. Talks about his friends in the field. And there's a chapter on his friend Werner Kirsten, professor of microbiology at the University of Chicago. And he says, my great friend Werner Kirsten leukemia virus, Kirsten sarcoma virus, found that he had to passage the leukemia virus in mice to keep it leukemogenic. And then one day he passaged it in rats to see what would happen. After a few passages, he took it back out of the rat, and lo and behold, it had a new property. It transformed cells in culture, and it caused fulminant erythroleukemia in the mouse. I read this, and I say, oh my God, look what happened. It picked up a rat gene. So we devised the right experiments, which took months to do at the time, and we found rat sequences in the Kirsten sarcoma virus, and we exploited the system for many years after that, and then I ended up going to Merck in 1982, because at the time the Cancer Institute decided, this is pre-Weinberg transfection experiment, that they really weren't interested in cancer virology. They weren't emphasized chemical carcinogenesis. They started reducing the size of my lab despite its productivity, and I decided I don't need this. And when I got an offer to go to Merck, and I had known Vagelos, I went to Merck. So if you want to know about Merck years, you can invite me back. So I think the take-home lessons here are, there's a lot of chance things that happen in your life, circumstances around you, but you have to have a kind of vision for what you want to do, pick an important problem, and figure out with the circumstances in your life that are available to you and that come up by chance, how you can pursue what your real emotional goal is. And you should never give up on that. And I do want to say one other thing about MIT, which I've said in other environments. I said that when I was at Harvard, and even subsequently we used to go back to Harvard, it was, perhaps not is anymore, but it certainly was a very different socio-cultural environment than MIT. When I was at Merck and would come to recruit at either MIT or Harvard, the people at Harvard always made me very uncomfortable, because despite what I had done in science before going to Merck, they clearly looked down at what I was doing at Merck. And I was never made to feel that way at MIT. Whenever I came to MIT, I was treated like a scientist and treated with respect. And it is a non-social hierarchical environment. The coin of the realm is what you do scientifically, not where you were born, not who your parents were, not how much money your family has, not where you came from. It's color of your skin, your blah, blah. And I have always cherished the place because of that. It's still that way, and you're really lucky to be students here. So thank you for inviting me.
I have to say my expectations have certainly been fulfilled, and I did learn a lot, and I did enjoy a lot, and I'm only sorry that we have to follow the clock. But I think even given that, I hope, Ed, you'd be willing to take a few questions at this point.
I'm free, so anybody wants to.
So,
Sure.
so questions, I mean, like,
Please.
How about the rest of your life? Okay, questions from the audience. Anything anybody would like to ask or hear more about?
Yes, please.
So what was the big difference when you went to Merck, all the way when you went from academia?
Well, the biggest difference, I didn't exactly know what I was getting into. Merck wanted to revitalize its virology department and was building a building and gave me a lot of resources, and I was very unhappy in Cancer Institute at the time. So I went, and I trusted Vagelos because I'd met him years ago. And after a few weeks, I realized what Merck was a big department of medicine focused on therapeutics, as opposed to just pathophysiology. And I loved that idea. You know, I had a good background in biochemistry and molecular biology and medicine. There were lots of illnesses and projects that I thought we could take on. And unfortunately at the time, Merck did not have the staff technical infrastructure, except a little bit in biochemistry, to do any of them. So it was a big recruiting process to try to get people with the right backgrounds in the right fields to come in and then start projects. That was the biggest transition. Yeah.
Yeah, one of the things that I loved about this talk is that you're kind of sharing your instincts in your nose to sniff out interesting new areas to get into. I wondered if you could just say something about how you landed in brain science in the Stanley Center.
Yeah, sure. As many of you know, some of you in the audience know, for very personal reasons. We have 3 children. Our second child, a boy, went to Harvard, extremely gifted, and developed psychotic illness after he graduated. And for many years, it's not clear what his diagnosis was, became clear in the late 90s that he had bipolar disorder and not schizophrenia. And for many years, between 1992 and 1999, 2000, when he became really, really sick, he became catatonic, and it had a terribly rocky road in between. Even tried to commit suicide at one point. I learned about the field both through his illness and volunteer work that my wife and I did for NAMI, the National Alliance of the Mentally Ill, how bad the field was. And I decided, gee, I'd really like to do something here. I was quite intimidated. But then I started reading journals and visiting schools to learn about neuroscience. And ended up, we don't have the time to go through it all, but ended up meeting David Altshuler at a meeting and ended up at the Broad to help them build their program. So it was really a personal mission in a sense. All I knew is that the genome had been sequenced. I knew enough to know that the field was hopeless unless we could find the genes, because you couldn't do biochemistry on living brain. And the Broad, Eric, David, were really good at it. So that's how I came here. And I was very fortunate that some people at McLean that I had been involved with, because my son had been hospitalized there for 6 months, put me in touch initially with the Stanley Medical Research Institute. And I went to visit them before I came here, told them, they had had a son with an illness, told them what I wanted to do, and they basically said, well, and I told them about my son, and they said, look, we see your track record, you're motivated, we'll give you some startup money. And that was the beginning of our interaction with the Stanley Institute. There's much more, but I can tell you in person if you want to know. Yeah.
I think with that, we should thank Ed very much.